Coming to my talk today. My name is Allison Kerwin. I'm currently a postdoc in the Medina lab over in Mueller, but today I'm actually going to be talking about the work I did for my PhD, which was at the University of Connecticut with Spencer Nyholm, and that was on this organism, Euprymna scolopes, the Hawaiian bobtail squid. So I'm going to talk first about the life history strategy of producing eggs. So eggs are a strategy for both vertebrates and invertebrates from a wide variety of different groups. Here's just a small sampling of those. And they're often unprotected for all or part of their development. And embryogenesis, or the length of time it takes for an egg to develop, can last for quite a while, so from days in some organisms to weeks or months, and in some octopus it can actually last years. So these eggs are quite vulnerable during this time period, due to predation, infection by various microorganisms, or by fouling. And so organisms have had to evolve strategies to compensate for these vulnerabilities, such as parental care. So this octopus is actually going to stay with her eggs right here until they hatch at the expense of her own health. They may produce numerous eggs in the hopes that some of them will make it to adulthood. And they may also use chemical defenses. And this can be either on the part of the host organism or of symbionts. And these defenses can be embedded in the eggs before they're deposited, or they can be produced during embryogenesis by either the embryo or by symbionts. So to narrow the focus just a bit, as I said, I'm going to be talking about Euprymna scolopes, the Hawaiian bobtail squid. And some of you may be aware that this has been a model organism for a number of years due to its light organ symbiosis with a bioluminescent bacterium, Vibrio fischeri. And that is a symbiosis that helps the squid to avoid predation. And it's located right here. This is a ventral dissection of a female squid. What you may not be aware of is that females also have a bacterial consortium in their reproductive system. And that's in an organ called the accessory nidamental gland, or ANG, right here. So if we zoom in a little bit, you can see these two large white nidamental glands, and then this really large colorful gland is the ANG. The ANG is conserved throughout many different squid and cuttlefish and was actually first described back in the very early 1900s, around 1908. But despite that, nobody's ever described a function for the symbiosis. The organ consists of a number of epithelium-lined tubules, and each one of those tubules contains its own dominant bacterial taxon. So you can actually see a couple of tubules here, and you see that they're different colors, and that coloration actually comes from the bacteria in the tubules. And if we look at this light micrograph, this cross-section, you can see this really thick epithelial lining, and then each tubule is just packed with bacterial cells. And there's usually on the order of about 10 to the 10th bacterial cells per ANG in a mature animal. The way the symbiosis works, so we have here the nidamental glands in the ANG. The eggs are stored in the mantle cavity, and when a female's ready to deposit her eggs, these nidamental glands secrete this really viscous material that we call the jelly coat. So you have this center yolk sac with the developing embryo surrounded by this thick membrane called the chorion. And then the nidamental gland extract wraps that center yolk sac in all of these layers of jelly coat, which you can see in this transmission electron micrograph. And then as that is happening, the ANG is going to secrete bacteria into these layers. So you end up with these single bacterial cells scattered throughout the jelly coat layers. And then the entire egg is going to be wrapped in an outer capsule, which is going to cement the eggs together into a clutch. So we know that both the ANG and the jelly coat are dominated by bacteria from the alpha proteobacteria class and from the Verrucomicrobia phylum. And we also know from some research I did earlier that the jelly coats will actually cluster in terms of bacterial community composition with the ANGs from the female that produce them. So I'm going to show a couple of these plots throughout my talk. So this is an NMDS plot. It's basically a way of visualizing how similar the entire community for a single sample. And the closer 2 points are to each other, the more similar that composition is. And then I've color-coded them by female. So there's 5 different females represented here. Diamonds represent ANG and circles jelly coats. And then the gray points are just provided in the background for context. They're tissues I didn't have the matching type for. And what you can see is that we have these clusters of an ANG with the jelly coats that that female produced. And so that shows us that the jellycoat community is mirroring those minor individual variations between females in their ANGs. So we also looked at how stable the community was once it was deposited in the jelly coat, and we found that there was no clustering of the communities by stage of embryogenesis, so it was pretty stable over the course of development. But we did find that they were metabolically active. So we saw an overall abundance increase of the bacteria. It started at about 10 to the 4th bacterial cells per jelly coat and ended around 10 to the 5th after about 21 days. So that's the duration of embryogenesis with these squid. And then we also saw that there were these small microcolonies that formed after 21 days of similar type cells. And we also found evidence of dividing bacteria at all time points. So it's an active community, it's just very slow in terms of cell division. So I'm going to first talk about how the ANG symbiosis develops, and then I'm going to come back to the function later in the talk. So the way the life cycle of these squid works, the eggs are deposited in rock and coral crevices. After about 3 weeks, the juveniles hatch, and their light organ is immediately colonized by Vibrio fischeri. And you might think, well, the ANG is probably immediately colonized as well. These bacteria are in the jelly coat. It would be very easy for the squid to do so. But the problem is the ANG doesn't actually develop and become available for colonization until about 1 to 3 months after hatching. So we believe they need to reacquire their symbionts from the environment. Animals will reach sexual maturity at about 2 to 3 months, and then they'll produce numerous clutches over their lifespan. And I'm going to be referring quite a bit to this metric called mantle length for the next several slides. It's just the distance from the base of the eyes of the squid to the back of the body. And size in these squid only loosely correlates with age. So you can have 2 squid that are the exact same age and they'll be very different sizes. But we do think that the size correlates fairly well with sexual maturity. So I'm going to come back to that a little bit. So ANG development has been studied in some other cephalopods, usually in either museum specimens or laboratory-raised animals, both of which present some issues. And usually these studies haven't extended across the full development. This is an image from one of the more recent papers from 1998, and this is a cross-section of the tissue. And in Doryteuthis, like in other cephalopods that have been looked at, we see this oh my goodness. You see these tubules forming through these ciliated invaginations, which then close off and have these tubules with bacteria inside. Okay, so I mentioned that using laboratory-raised squid is a problem. It would be great if we could, because then you wouldn't have to worry about this loose correlation between size and age. You would know how old the squid were. And so we've worked on this in our laboratory, and this is again what a wild mature ANG looks like. So we raised a number of squid to adulthood, and what we found is that in about 80% of the time, when females were raised in laboratory conditions, there was no ANG present at all in the adults. It was completely absent. And about 20% of the time, we saw this small stunted ANG. These were usually, as I said, very small, they lacked color, and they tended to be asymmetrical as well. They had very low alpha diversity, so this is looking at metrics like the richness of the bacterial community, how evenly distributed those bacterial members are, and phylogenetic diversity of the community. And they had a very highly variable community composition. So you could have 2 squid that were raised together, and one would have, say, 90% alpha proteobacteria, and one would have 60% flavobacteria. So it didn't really make a whole lot of sense in terms of a consistent community. So our next step was to actually bring in wet Hawaiian sand, which we did. The post office must have loved that. So we would raise the squid on that. And what we found is that when we did that, we actually got ANG morphogenesis. So these ANGs are still stunted. They're not as large as we would see in the wild squid over here. But we do see distinct tubule formation. You can see here these twisty-turny guys. And there is some coloration present as well. They also weren't asymmetrical anymore. So we're definitely improving. The richness and evenness was actually rescued by raising them in these conditions, so it returned to the wild ANG levels. The phylogenetic diversity went up, but it's still not quite at those wild levels. And you can see in this beta diversity plot, so These are animals raised in Connecticut under laboratory conditions. Wisconsin, the conditions are a little different, but I'm not going to get into that. And then the blue are the wild. And then you can see that these Connecticut animals raised on Hawaiian sand are much closer in community composition to the wild squid than we've seen previously. We've also recently shown that you can actually add specific isolates to these raising conditions if you're using the wild Hawaiian sand, and the squid will incorporate those isolates into their ANG, and you can recover them from the adult ANG. Okay, so why did I go into all of this? Well, it's to say that it's still a little dicey to use these in development stories because we're just not sure exactly what's happening here yet. But I am going to use, for some of the smaller time points, these animals that were raised on Hawaiian sand because we haven't been able to obtain those samples from the wild. Okay, so I'm now going to take you through what we think is happening with the ANG as these squid develop. So at the smallest time points, at about 4 millimeters, that's the earliest we've seen any evidence of an ANG, we see this really small nub of tissue, and there are all of these channels running throughout it. At 5 millimeters, you can sort of see that it's spread out a little and there's almost 2 different planes present. And if we look at a transmission electron micrograph, you can see these channels, and I'm going to show you some better TEMs in a little bit, but they're actually coated in cilia. By the time you reach a 6 to 10 millimeter mantle length, we can see this really distinct bilobe structure, and those channels are still present around the outside of each lobe, and then the center is covered in all of these pits, which you can see here. And again, we see a lot of cilia present there. So these are all animals that were raised in the lab, and I'm now going to switch over to only using wild animals for the rest of the talk. So this is at about 11 millimeter mantle length, and you can see that the ANG has formed this really interesting structure that it maintains in a similar way up until about 13 millimeters. And there are these 2 lobes, which in the smaller animals are sort of coming together and pushing out. So you have this really large surface area. And these structures are completely covered in these pits all over the surface of the nascent ANG. And we counted, and there's about 1,000 of these pits present on one of these nascent ANGs. So there's a lot of surface area and a lot of these pits. So what's going on at the cellular level when we look at this? Well, if we look at these light micrographs, you can see that there are actually sort of 2 layers of tissue at this stage. So this is at about 13 millimeter mantle length animal. In the surface, we have these ciliated invaginations. That's what all of those pits are. And they're just all over the surface of the organ. If we go into the more basal layer of tissue down here, you can see structures that are more similar to the tubules we find in adults, as well as a lot of connective tissue. And if we look at a transmission electron micrograph, you can see that the entire surface of the ANG is actually covered in microvilli and cilia, which actually continue down into those invaginations. And we actually don't find any bacteria at the surface of the invaginations, but if you trace these invaginations down to their base, or similar to a crypt in the light organ, you'll start to see maybe 1 or 2 bacterial cells there. And these structures, having these microvilli and cilia present, are very reminiscent of what we see in other organisms that have tissue that are poised for colonization by symbionts. So in deep sea mussels, if you look at the gill tissue, it's continuously generating new gill tissue that needs to be colonized by their symbionts. The new tissue will have microvilli and cilia on the surface. Once that colonization occurs, those structures are lost. So we think something similar is going on here, and these ciliated invaginations are poised for colonization by the symbionts. If we look at the basal layer of the tissue that I mentioned, we see these tubule structures similar to what we see in adults. They do contain bacteria. There's not quite as many as we would see in the adult tubules. You'll see that later on. Whoops. Oh, sorry. I thought the arrows weren't up yet. So these white arrows are pointing to some cells that are actively dividing. And we generally saw cocci in most of these tubules at this stage. And some previous work from our lab by another grad student, Andrew Collins, associated these cocci with the Verrucomicrobia contingent of the population. So we think we're seeing mostly Verrucomicrobia at this stage. I'm going to come back to that in a little bit. Okay, so then we're going to go from this bilobe structure. Those two lobes are going to merge together into a single lobe. And it's not a great image, but you can see there's sort of one single piece of tissue there. There's some coloration in the center. And we're seeing distinct tubules form at this stage. So we're at about 15 to 17 millimeter mantle length. And at this stage, when we look at the cellular level, we see that it's just completely changed. So we no longer have that surface layer with all those ciliated invaginations. There's no more cilia or microvilli on the surface at all. So that window for colonization seems to have closed at this point. And we're only in a 16 millimeter animal, so it's only 3 millimeters larger than the last set of images I showed you. And so the entire tissue is now made up of these tubules that are just completely packed with bacterial cells. And really this looks very similar to what we would see in a mature animal. So this is a sexually mature animal that was greater than 20 millimeters. So it's just an entire organ made up of these tubules. So then after that stage, you're going to just see more and more color. The ANG is going to continue to grow. You'll start to see a couple of eggs in the mantle cavity. And by the time you hit 20 millimeter mantle length, you have, in pretty much all cases, a fully mature squid. So the progression we're seeing is that you start off with this small, tiny piece of tissue that's got these channels. Eventually, those are going to migrate a little bit to the outside, and you're going to start to see pits in the center, those ciliated invaginations. And then you're going to have those grow until you have this really large structure with a lot of ciliated invaginations ready for colonization. And then once that colonization occurs, you're going to switch over and become an organ that's just composed of the tubules instead. Okay, so I've spent a lot of time talking about the organ, and we're here at the Microbiome Center. So what's happening to the bacteria while all of this is going on? So to look at this, I did next generation sequencing of the V4 region of 16S rRNA. And here I'm going to show you these different groups of bacteria plotted with mantle length on the x-axis, relative abundance on the y-axis. The red line is at that 20 millimeter point. It's just to orient you about where we expect maturity to have definitely occurred by. And then I've plotted either a linear regression or an exponential curve to the data, depending on which one fit better. And what we see here is that the Verruca microbia are exponentially decreasing as you move towards maturity, while the alpha proteobacteria are exponentially increasing. And so we're moving from a community dominated by Verruca microbia to one dominated by alpha proteobacteria. At the same time, we're seeing a linear increase in the flavobacteria levels, and the gamma proteobacteria, there's a slight upwards trend, but they're really staying pretty stable over the course of development. All bacterial classes that we associate with the ANG are present in the earliest points that we're able to sequence these communities. So I'm starting at 10 millimeters. I did show you that the ANG is present before that, but there's so few bacterial cells there at that point that we can't really get useful sequencing out of it. And so what we think is going on here, I've shown you all of these images of these ciliated invaginations, that there's probably about 1,000 of them. And so we think the Verruca microbia may be much better at colonizing these invaginations. So they come in and they colonize the majority of those available spaces. The alpha proteobacteria, maybe not so great. They come in and they colonize a few here and there. But at that point, and so at that point, you're going to have a relative abundance that's very dominated by Verruca microbia, even though there's not a lot of cells present yet. But then the alpha proteobacteria tend to be very fast-growing organisms. So under laboratory conditions, they have a doubling time of about 4 to 6 hours for most of the groups that we find in here that we have in culture. Whereas the Verruca microbia tend as a group to be very slow-growing organisms. So we don't actually have the Verruca microbia for this system in culture yet. We've been trying for a long time and haven't succeeded. It's still ongoing, but in most of the literature we see that it can take a month for Verruca microbia to form colonies on plates, so very slow growing. And so I think what's happening is the alpha proteobacteria tubules just divide and end up taking up much more of the eventual volume of the ANG than the Verruca microbia tubules. So that's our working hypothesis for right now. We also looked at how alpha diversity was changing with maturity. And so again, have the mantle length on the x-axis. This is a metric called the Shannon index, which measures both richness of the community, or the number of species present, and the evenness, so how evenly distributed those species are. And you see that there is a significant increase, a linear increase as you move towards maturity. When we looked at phylogenetic diversity, we see that there's a slight upwards trend, but it's not significant, so this is pretty stable. And we also looked at a few other metrics, and we think what's actually increasing here in the Shannon index is the evenness of the community. And so the Verruca microbia in this system is dominated by pretty much one or maybe two OTUs. So there's only a couple of species present there. And so you start off with this community that's dominated by basically one type of bacteria. The alpha proteobacteria, there's a lot of diversity there. There's a lot of different families, genera, and strains, which you're going to see in a little bit. And so you're going from a community dominated by one type to one dominated by a lot of different types. And so the evenness is really increasing as you move towards maturity. And if we look at a beta diversity plot here, I've sized the points by how big the animal is. And you can see that there is a correlation. So squid that are less than 18 millimeters cluster to the left of the NMDS plot, whereas there's really no signal once you hit that 18 millimeter point. So we think that once you hit 18 millimeters, the ANG can be considered a mature organ. The community composition is mature, the cellular structure is mature. It seems like that's where that maturity point is. So to wrap this section up, we've shown that the nascent ANG is primed for colonization with these ciliated invaginations. We're seeing a shift in the dominant taxa of the ANG and an increase in the evenness of the community. So we're really hoping to continue to try to understand the dynamics of this early ANG colonization. And we also want to start looking at whether the tubule microenvironments could possibly be changing to foster the growth of certain taxa. So in the light organ, you have this trafficking of the hemocytes, which are the immune cells of the squid, in and out of the crypts. And when that happens, they actually contribute chitin, which leads to increased oxygen availability in those crypts and acidification. And that favors the growth of the light organ symbiont Vibrio fischeri. And so it's possible something similar could be happening here. We also see hemocyte trafficking in and out of these tubules in adult animals. So there could be something similar going on, and it's something that we're interested in exploring more in the future. So we want to continue to compare the mechanisms of ANG development to that of the light organ. Okay, so I've talked to you a lot about the symbiosis and how it develops now. Why are we so interested in it in the first place? So squid, like many other aquatic organisms, lay their eggs in seawater, and seawater, as I'm sure you guys know, has over 1 million microorganisms per mil present. So in this type of an environment, biofilms can form incredibly quickly. But squid eggs are not subjected to any fouling or predation during development. So I've said that in Euprymna, this development period lasts a little over 3 weeks. In other squid, it can be a little bit longer, but nothing really touches these eggs, even though they should theoretically be a good source of nutrients for various fouling organisms and predators in the ocean. We also know that some marine bacteria, including some that we associate with the ANG, are able to inhibit fungal, bacterial, and algal growth. And so there's been this hypothesis floating around since about 1991 that these jellycoat bacteria could protect the squid eggs from fouling and predation. However, nobody's published any experimental evidence to date that shows the function of this community in vivo. There have been a few studies that have looked at the in vitro activity of some isolates, including some that have come out of our lab using just in vitro assays, and they've found some antibacterial activity. So I took clutches and I treated them with antibiotics, and that was either chloramphenicol or an antibiotic cocktail, and that reduced the bacterial load of the eggs by 98%. And when we did that, the eggs grew this incredibly large biofilm over the entire surface. And when we looked at the biofilm under the microscope, we saw these very characteristic fungal hyphae, as well as a number of bacterial cells scattered throughout. And this biofilm formation killed the embryos across the board. There was very low viability in these cases. Like I think it's around 3%. And we actually were able to then identify this fungus as Fusarium keratoplasticum. And this is a very ubiquitous marine and terrestrial fungus. It can infect a wide variety of hosts, including people. It's actually a big problem in certain regions. And we then repeated these experiments in flowing Hawaiian seawater in Hawaii and found similar results, showing that this could be a threat to these eggs in their natural environment as well. So we usually saw the biofilm appearing around days 8 to 11 of embryogenesis, so about the halfway point. I wanted to look at this in a little more detail. So in these images, I'm going to show you a close-up of the surface of the eggs. So in this first image, this is one egg down here connected to another egg up here. And you can see that as early as day 3, we're already seeing some fungal hyphae appear on the surface of this antibiotic-treated egg. By day 6, you can see these hyphae have really started to spread across the surface. And by day 8, there's pretty much a strong biofilm. This is actually the same grain of sand on these 2 eggs, which was pure luck, but you can see how much this biofilm has grown in just 2 days. By day 10 and 13, it continues to spread, and by day 15, it's completely taken over the clutch. So we know that bacterial protection from fungi has been documented in several different invertebrate systems. In both shrimp and in lobsters, the embryos are coated in bacteria that can produce compounds to inhibit the fungus Ligenodium calinectes. And in hydra, if you treat with antibiotics to remove their epithelial bacterial community, they also develop a fusarium infection. And in hydra, you can actually then complement with their normal bacterial community, and they're able to then fight off that infection. So based on this evidence and these antibiotic experiments, it really looks like the bacteria in our system are preventing fungal fouling. But we wanted to begin to tease apart the effects of the bacteria from potential host factors. And so to do that, I did this experiment where I divided the egg into different components. So some eggs I left intact. Some we removed just the very outer capsule of the egg, leaving the jelly coat and the bacterial community intact. And some we have just the yolk sac, so the jelly coat has been removed as well. And then I challenged these different components with a suspension of Fusarium keratoplasticum bud cells. And in these images, I'm going to show you eggs that were not treated on the left and eggs that were challenged on the right. And 10 to the 4th is sort of the maximum fungal load you'd expect for seawater, but it's going to be in the wild a mix of different types of fungus, whereas here it's a monoculture. So this is well above what they would normally be experiencing. And there's no antibiotic treatment in this. This is just the normal community function. Okay, so whole eggs, when untreated, as you would expect, develop fine. There's no fouling on the surface. When we challenge these eggs with the 10 to the 4th suspension, and this is after about 18 days of development, so pretty close to the end, we do see a few fungal hyphae appearing on the surface of the eggs, but it doesn't ever develop past that about day 3 biofilm I showed you a few slides back, and it wasn't enough to harm the embryos, so they developed fine. When we remove just the outer capsule, so the jelly coat is still intact here, and left it untreated, again, it developed fine. And again, when we challenged it, we see a few fungal hyphae appearing here and there, but again, the embryos developed fine. So then we removed the jelly coat as well. So now we just have the yolk sac with the developing embryo inside. And again, when left untreated, they did develop fine, although they were extraordinarily fragile at this stage. And when we challenged them, they developed this incredibly thick biofilm that completely took over the eggs, killed the embryos. And when we looked at this under the confocal, stained with the nucleotide stain Cyto9, you can see just this mass of fungal hyphae present here. So this experiment, along with our antibiotic experiments, really showed us that the jelly coat and its bacterial community were crucial for defense of these eggs against fungus in this system. But we wanted to go a little further and start looking at what these eggs could possibly be producing to prevent fouling. So we obtained a lot of different isolates from either the ANG or the jelly coat. You can see that we're a little biased towards alpha proteobacteria, and as I mentioned, we're lacking the Verruca microbia. And then another member of my lab, Andrea Surya, performed inhibition assays. So in these assays, she cut wells in agar plates, and she would fill those with whichever isolate she was interested in, whichever bacterium, and she would let that grow for a couple of days, and then she would add fungal bud cells and let that grow for a couple of days. And you can see in the negative control, you would get this large mass of fungal hyphae. This is a positive control with the, I think it's cyclohexamide, which kills the fungus, and so you get no growth there. And so here are 2 isolates she did. These are both Leysingeras, which are alpha proteobacteria. And you can see that this isolate has completely inhibited all fungal growth from occurring, whereas this one, there's been some growth, but it's still well under the normal amount you would see. And then here is an isolate from the Alteramonas, a gamma proteobacteria. And in one case, there's been very little to no inhibition, and in this case, almost complete inhibition. And I'm just throwing this up there to show you, this is the only difference between these two plates is that one was done with glycerol in the media and one was done without glycerol. And this is just to point out that the media you use can really affect results in this type of a study, so you want to be aware of that. We actually did all of these assays both with and without glycerol, and it's not a trend where glycerol always reduces inhibition. It's variable, so it depends on the isolate. We then collaborated with a lab in the pharmacy department at UConn, and they obtained a number of organic extracts from these isolates and did plate assays. And this is the Balloonist lab, and this is work done by Samantha Gromick. And in this heat map, I've got the extracts sorted by isolate type. So these are all the alpha proteobacteria. I've got the controls down here at the bottom. We tested against 3 strains of Fusarium and the human fungal pathogen Candida albicans. And the yellow indicates that there was complete inhibition of the fungus, whereas the blue indicates no inhibition. And you can see first, right off the bat, that there's a lot of yellow. So a lot of these extracts were able to inhibit the fungus. But what I found really interesting here is that we would we would frequently see extracts that were able to inhibit all of the Fusaria, but not the Candida, or the Candida and none of the Fusaria. And so there seems to be this really strong differential activity of these different extracts from these isolates. So to wrap this section up, we've shown that the jellycoat bacterial community is crucial for defense against fungal fouling in this system, and that these isolates are able to produce organic compounds, probably secondary metabolites, that can differentially inhibit the fungi. We've started working on identifying some of these active compounds. This is work going on in the Balloonist lab. Some of the ones they've identified so far are mycinomycin, which has some anti-fungal activity, and these glycerophosphocholine, primarily lyso-PAF, which we're also working on now, but we're going to continue moving forward on that. And in the future, we'd really like to do some complementation experiments where we try to add back in bacteria or extracts that had these anti-fungal properties to try and protect eggs that lack their normal microbiota. So I'm going to wrap this up just by showing you this model we've come up with for egg defense, which is that different bacteria in this complex community have slightly different roles. So I've hopefully convinced you today that if you have fungus coming in and trying to foul the eggs, you'll have a subset of bacteria in the community that are able to produce these secondary metabolites to prevent fungal fouling. We've also got evidence from our lab that a similar antibacterial role is played by another set of bacteria in the community. And we think something similar could be going on to prevent larval settlement on these eggs, and then possible algal spores from coming in and fouling the surface of the eggs as well. There we go. And I don't want to leave you with the idea that there are no host factors that work in this system at all. So there's a paper from the 70s that shows that the nidomental gland material that forms the jellycoat actually can inhibit the ciliary bead of certain protists. So the jellycoat itself may be able to prevent protists from coming in and infecting the eggs. But all in all, you have this really interesting dynamic system where these bacteria have these very specific roles that are maybe slightly functionally different, but have the common goal of protecting the egg. And this has really been a great project because it's shown us that if we integrate our knowledge of community composition with some functional ecology and natural product discovery, we can really begin to tease apart the roles of the different members of a symbiosis. So with that, I would like to thank my current lab, the Medina lab, my former lab, the NIHOME lab. You saw work from Andrea and Sarah today, and then Joey and Jess are 2 undergrads that worked with me. We had a number of collaborators all over the place, and our funding was from NSF, UConn, and experiment.com. And with that, I'd be happy to take any questions. Yeah, so I think there could definitely be some competition aspect to it. But the bacterial load of the eggs isn't that high, so it's about 10 to the 4th to 10 to the 5th, depending on where you are in development. So they are pretty scattered throughout the jelly coat. So it seems like the secondary metabolite production is probably playing more of a role. But I do think that there is probably, and so part of your question was, do we see the same compounds from all of these? And the answer is no. So we've been doing, I didn't show it today because it's still sort of in preparation from the other lab that we're working with, but they've been doing molecular networking analysis with the mass spec data they've obtained and looking at which compounds are produced by which members of the active, the anti-fouling active isolates and extracts versus eggs that were challenged with fungus and all of these different combinations. And so we are seeing different compounds in different ways. So I think there are some different ways that these isolates are defending the eggs and, well, themselves from this fungal fouling.
So I have 2 questions.
Yeah.
First one, have you done fish with the developing animals? So do you see displacement of the verrucomycetes?
The verrucomicrobia, yeah.
Yeah, the verrucomicrobia with the alpha in the same location, or do you see that the alphas are just colonizing elsewhere in the ANG?
So we haven't done fish of the developing animals. So one of the problems we've had with a lot of this work is that we're really sample limited. And so most of our samples went to the sequencing effort. And now that we're using some of the raised animals, we'll have a little bit more in terms of sample size for some of these smaller animals. But we've started more with confocal microscopy and not fish. But I think that's something in the future. We do know from work that Andrew did that in the adults, each tubule only contains 1 taxon of bacteria pretty much. So like it'll be 99% Lacingera. And then another tubule next to it will be 99% Verrucomicrobia. So it does seem that each tubule is only colonized by 1 primary group of bacteria. So for what it's worth.
Okay, and so my other question is, I guess with our squid facility, what we end up seeing is as the animals age, the adults age, the viability of the egg clutches decreases. And so eventually we're at this point where some of the squid are still laying clutches of eggs, but we get maybe 2 or 3% of the animals hatching from those clutches.
Right.
So we had just speculated that it was all due to the age of the animals, the egg, I guess the embryo's quality going down.
Right.
I'm just curious, do you think perhaps we're losing the diversity of the ANG in the adults, and then we might be losing viability due to that lower diversity of the jelly?
So my short answer to that is no. So I did a study that I didn't show today where I looked at animals that were brought in and maintained in the lab for various points of time. I think the longest was about 4 months, which is, I don't know about you, that's the max we usually have animals for. And there is no change in the community composition over that time period. So it doesn't seem like the ANG is being affected by bringing animals into the lab at all. So I don't think that's happening. We also never really see on our normal eggs that we keep in our facility any kind of evidence of fouling, even on the later stages, although we do see the decreased viability you're talking about. So I don't think it's related to this. So the raising, we're starting to do F2 generations and possibly F3. And so there's been some anecdotal evidence from the Monterey Bay Aquarium that did these experiments that viability also decreases with generation. And so that I do think could be affected by the ANG community because they have to reacquire it from the environment. If the normal bacteria aren't present, then they're not going to have that same function. So they'll be more susceptible to things. Yeah.
Just to that last point, I think the aquarium, though, doesn't it use unfiltered water?
It does. Well, I don't know if it's unfiltered. It might be like coarse filtered, but it's not, yeah, like not UV or anything like that.
So it's not, I mean, it's not lab water.
Right, it's not lab water for sure. Absolutely. But it's not, Hawaiian water either. So these squid are, so there could be a different community there. I don't know enough to really speculate on that. I think that's why they are seeing ANGs in their animals. We wanted we tried for a while to get them to send us their females when they would die, because I really wanted to see what the ANGs looked like, and they had been going for I think 8 generations at the end, and they never were able to send us any. And the person that was doing those experiments is now at Woods Hole, so it's ended there, so we don't have a chance to look at them, but it would have been really interesting. So we're hopefully going to be able to replicate some of that in the lab.
So just another kind of comment and something, I think there's a number of people in plant pathology that would be happy to sit down and talk with you about coffee. Is that we, in plant pathology for years, we have done experiments looking at secondary metabolite production and biological control, and the difference between what we see in vitro and what we see in field studies.
Right.
And we have a lot of information about how and when we can correlate material or information we see from the lab.
Right.
So, for example, your glycerol triggering secondary metabolite effects, let's say.
Yeah.
But there's a whole literature about organisms in secondary metabolite production and how to even study that in situ that would be interesting for you to know about or to share with the lab.
Yeah, definitely. I know Andrea she's the one that's done most of those experiments, and she's definitely looked into a lot of that, but I'd love to hear more resources.
There is a person who is one of the world's experts in this field coming for the phytobiome.
Oh, okay, right, yeah.
Symposium this summer in June, and he would be an excellent.
Oh, that would be great. Yeah, definitely. Thanks. Yep.
So your lab-raised squid that have these very small or non-existent ANGs. Do they still produce eggs?
They do, yes. So we have been able to get them to produce eggs even without ANGs. The eggs do have some bacteria in them, which I've sequenced, and those communities tend to be all over the place. And I sort of think what's happening with that is that as water is flushed through the mantle cavity and they're coating these eggs in the jelly coat, it's very sticky. Even when we go to do dissections, it's this really sticky material. It's a pain in the neck to work with. And I think that any bacteria that are sort of floating around just sort of get trapped there if the ANG isn't there to really like deposit a lot of bacteria in. So one of the experiments we really want to do is raise these squid without ANGs and then test those eggs and see if they get biofilms or not. We transferred from doing those experiments around the time we were starting to think of that idea to using the wild Hawaiian sand. And so Sarah McAnulty from my lab is going to go back and do some on lab sand and see if she can do those experiments, but they're a little ways down the road because we're trying to, it's hard to have both of them going at the same time and not worry about cross-contamination and that kind of thing, so, yeah.
And so I thought you also suggested that if you add other bacteria in, they can get incorporated in. So now, is there any evidence of selection by the host for desirable organisms?
I would say that the evidence for that is that it's a very stable community across populations of squid. I've done 2 different populations that are genetically isolated in Oahu, and then multiple generations of squid. So over about 5 or 6 years, these squid live about a year in the wild. And we've seen pretty much the exact same community composition in all of them. So I think there is evidence that it's a very strongly conserved symbiosis. But in terms of how they're selecting or any other information like that, we don't have that. We also know if we add isolates to lab sand that we know are part of the community, we don't see ANG formation. So we've done that experiment with a few different types of alpha proteobacteria, some gammas, and some flavobacteria, and we have not seen ANG morphogenesis with any kind of regularity. We do need the wild sand, so we think the Verruca microbiome may be the key to this because we don't have them in culture yet. And so we think that when we bring in the wet sand, it's got the Verruca microbia present in there, and that's what's causing it. And so, yes, I have.
Identical composition between what you get in the ANG? Because I mean, that would
no, absolutely not. So we've sequenced sand from the wild in Hawaii, from the environment where these squid live. And then I've sequenced the sand that's brought into the lab and then done that over time as we keep it in the lab for a year. The community obviously is going to change a bit. And then also the sand just from our lab. And the Verruca microbia is present at very low levels in the sand. It's at maybe like 0.1 to 1% max. So it's really low. And so we're seeing a significant enrichment in the ANG as well, which is similar to what we see with the Vibrio fisheri as well. So it's present at very low levels in the environment, but it obviously is the only thing that can get into the light organ. So, yeah.
The fusarium was getting introduced from the Hawaiian sand as well?
No, I do not. I think the fusarium, for most of my experiments, is something that is just present in the building. So fusarium is really an obnoxious fungus. It grows in.
Wait, we love fusarium.
Okay, well, that's fair, but I don't know that you necessarily want to go around breathing it in either. And during a lot of my experiments at the beginning with the antibiotics, we had this really old air conditioning unit in our squid room. And we finally got it removed because it was nasty and we didn't want it anymore. And they put in a new one. And we started seeing lower levels of infection at that same time. So I'm pretty convinced that it's just a member of the, plumbing system in the building that we use, which is why you also see this in Hydra. So those are lab experiments as well. They get these fusarium infections when you treat with the antibiotics there. So I think it's just a really common member of these, the air and the plumbing systems in a lot of places. So that's why I did mention that we did these experiments again in Hawaii. They were not in the field, they were in a marine lab there. And we were doing pulse antibiotics so that we could then put them in the seawater, the running seawater tables. And we saw the same infections with it wasn't the same strain of fusarium, but it was closely related. And so one of the things I'm not going to be able to do, but I want somebody to do in the future is to do pulse antibiotic treatments on the eggs and then put them back out in the ocean under cages and see if they get fouled or not, because I think that would be really fascinating.
So he's undercover from the Fusarium Research Center. I just want you to know. I just didn't want to stepping into it. It works actually on organisms that attack animals and sea animals and drains that colonize drains.
Yep. Yeah.
All right. Thank you.
Thank you.